Hello, Mr. Allman here, and this is your HL Allman cast on genetics. In this, I'm going to be looking at a couple of different things on two different prezzies. The first is going to be on the Intro to Mendelian Genetics prezzi, looking at the classical dihybrid cross. The second is going to be extensions to Mendelian genetics, cases of recombination and gene linkage, and how it is that that impacts inheritance patterns. So let's look first as a reminder. Law of independent assortment based on Mendelian genetics is that separate alleles for separate traits are passed on independently. So in the case of the pea plant, if I have a trait for plant height and a trait for flower color, each of those is going to be passed on independently. And so whether or not a particular plant passes on a trait for purple flowers and short versus white flower and tall, though the choice for which allele is going to be passed on has nothing to do with one another. And that has to do with the fact, under the assumption that they are located on separate chromosomes. So in this example here, we see that there is two different traits being tracked here. We have the Y trait, which is looking at the color of the pea, and the R trait, which is looking at the round versus wrinkled. And so we can see yellow, round are going to be the dominant traits, and the green and wrinkled are going to be the recessive traits. So in this case, we have the P generation. If those are true breeding lines for each of those sets of traits, they create an F1 generation that's going to be what's called a dihybrid. A dihybrid is going to have a combination of, is going to be heterozygous for both of the traits being tracked. So heterozygous for color and heterozygous for shape. So when this individual produces gametes, those gametes can have either the dominant or recessive trait for either of those two alleles, because those genes are located on two different chromosomes that are going to line up and segregate independently of each other based on that law of independent assortment. So when we do the cross here, we can see that because of independent assortment, that we have four different possible combinations. We can have dominant-dominant, recessive-recessive, or either of the two combinations of one dominant and one recessive trait. When we're crossing individuals that are both heterozygous, that's how we get each of those four different possible combinations. So one thing to make note of is that in each case, the gamete in the Punnett grid, we are putting two different alleles, one allele for color and one allele for round. That means that when we put it together and we see what is the possible genotypes of the offspring, each of those is going to have four letters, two alleles for each of the two traits, so four total alleles. Now the phenotypic ratio, this is something that is important for you to understand, and it's good for you to remember, is 9 to 3 to 3 to 1. So in this case, how many of the offspring are going to show dominant for both traits? So how many are both round and yellow? So you can see that there's 1, 2, 3, 4, 5, 6, 7, 8, 9. So that's 9 out of 16 are dominant for both traits. How many are going to express one of the dominant traits but not both? So those would be yellow and wrinkled, 1, 2, 3. Green but round, 1, 2, 3. And so there we get 3 out of 16 for each of those. And there's only 1 out of 16 that will be recessive for both of those traits. So phenotypic ratio, the 9 is dominant for both, the 3 is dominant for one, recessive for the other, and dominant for the first, and then the 1 is going to be recessive for both. Now, if the genes are linked together, if the offspring really are passing on the traits with each other, either dominant with dominant, recessive with recessive, that greatly reduces the amount of diversity. So if independent assortment is true and they are located on separate genes, then you're going to see greater diversity in the offspring when you actually cross those heterozygotes with each other. Looking ahead, a Punnett grid can be used to be able to track the outcome of a cross between two parents when tracking two different traits. Use the Punnett grid only when the genes are unlinked, so when they are on separate chromosomes, and independent assortment is going to happen, then a Punnett grid is useful. In this case, you need to combine together whatever the possible combinations are. And so here in this case, if we have an individual that is heterozygous for both, the two parents have the same genotype as each other, and so we're going to see identical gamete combinations on both the top and the side of the Punnett grid, and then filling out that Punnett grid and then analyze it from that point. It's time consuming, but again, if you remember that ratio 9:3 to 3:1, which again applies anytime we are crossing two, we're doing a dihybrid cross, meaning two heterozygous individuals crossed with each other, you can save time and skip directly ahead to determine the genotypes based on that 9:3 to 3:1 ratio. Math is also something that you can apply here to genetics cases, particularly if you get a situation that is more complex, because what is going to be true when independent assortment is true is that the likelihood of any one given phenotype is going to be a result of the combined probability of each of the individual events. So you can read through what's on the screen there. But if we look at an example, here you can see if you analyze each trait independently, so for example, if I have the case here of yellow versus green, what is the probability of getting an individual that is yellow versus green? Well, 3 out of 4 yellow and 1 out of 4 are going to be green, because 3 out of 4 are going to have that dominant trait. Similarly for round versus wrinkled, 3 out of the 4 are going to be round, 1 out of 4 is going to be wrinkled, and that's something you probably don't actually even need to do the Punnett grid to figure out. So if a question is, what is the probability of pea plant being both yellow and wrinkled? Well, there's a 3 in 4 chance of it being yellow, and there is a 1 in 4 chance of it being wrinkled. And 3 out of 4 times 1 out of 4 is going to be 3 out of 16, and now you know that automatically there's a 3 in 16 chance of an offspring having that particular trait. So sometimes that will help you, again, if you don't need to know all of the possible combinations, but if you're just looking for one particular trait, using probability is going to save you quite a bit of time. This could also be used as a way to be able to figure out when we go to an even greater number of traits. You could do a tri-hybrid cross or quad hybrid. You could go to as many as you would like, but in each case you just need to multiply the probabilities of individual traits by each other to get the overall probability of a particular combination. Here's a couple of examples here. So you can see in this case we have the normal versus albino, tall versus short, yellow versus green, round versus wrinkled. And so if you'd like, you can do those crosses and see what is the genotypic and what's the phenotypic. Usually phenotypic ratios are going to be more significant when dealing with multiple traits of the F1 generation. That's going to result from the crossing of these hybrids, or the crossing of, on the right, this case, a hybrid with a homozygous recessive. I've moved over now to the extensions to Mendelian genetics Prezi. Again, remember that there are two different presentations that I'm utilizing to produce this particular video. So if we look at the idea of dominance, again, Mendel was using the pea plant, which is more of a model organism, meaning it's a little bit simpler, and as we look at human genetics and many other species, that dominant-recessive relationship is not something that we see happening commonly. That's more the exception than the rule. So some other types of relationships that we see, one of those is incomplete dominance. And so in this case, if we have a heterozygous condition, it's going to make a new third type of phenotype. This is not quite the same as the idea of blending, but it does incorporate a little bit of that concept. If we look at a snapdragon, in this case, this flower, if we have a parent that is white and a parent that is red, we are not producing a red or a white, but we are making a new color, a phenotype that was not present in either of the parents. So this is a case of incomplete dominance because both of those parental alleles are expressed. The whiteness and the redness are both expressed in the offspring, but in their expression with each other, they are producing something that is new. This is going to alter the ratios because as we look we see that there is 100% of the F1 generation is that new pink color. And now as we look at the phenotypic ratio, we're not going to see a 3-to-1 situation here as we would have expected if it was strictly dominant recessive. What we're going to see is a 1-to-2-to-1 ratio, because those that are heterozygous have that new phenotype, and so not the genotypic but the phenotypic ratio, and actually both of them ratios are 1-to-2-to-1. Codominance is going to be a condition that's going to result when both of the traits are going to be expressed simultaneously. This is going to differ from incomplete dominance, where two alleles were both expressed to create a new phenotype. In this case, both of those alleles are expressed at the same time, not creating something new, but creating a situation in which both are going to be expressed. A more visual example I can give you first would be an example of a spotted animal. So if we look at, for example, a black dog and a white dog that mate together, if the fur coat is expressed under a codominant inheritance pattern, then you're going to see the offspring that are going to be both white and black spotted. They didn't blend together to make a new color, but they express both the whiteness and the blackness in their fur coat as a result of the fact that both of those alleles were expressed. An example that's going to be significant to understanding immunity and how blood type is going to be inherited is how blood type and how those A and B traits are actually codominant with each other. So in the case of A blood, A has one particular appearance of its blood cells, B has a separate appearance of its blood cells, and they are codominant with each other. And so that's how we can get a phenotype of AB blood. Someone with AB blood type is really actually expressing both the allele for A blood and the allele for B blood simultaneously. This is going to have an impact quite a bit in terms of understanding how blood transfusions and interactions of blood type, and that's something that we'll cover more in detail in our unit on immunity. Other cases, when we see that there are more than two choices, it's not a dominant or recessive, expressed or not expressed, and this is going to be true again for many phenotypes which are influenced by multiple genes in different spots. So first we have multiple alleles. So there can be more than two alleles for one particular gene. So in the case of blood type, there are 3 alleles, the allele for A, for B, and for O type blood. And so we need to accommodate for additional phenotypes based on the fact that we have more than 3 possible outcomes. We don't just have homozygous dominant, heterozygous, and homozygous recessive. If you have 3 or 4 or 10 different alleles, there are many new combinations of alleles that you could then have, creating new phenotypes. Polygenic inheritance is going to be when there is more than one gene that contributes to a particular trait. So human skin color, human eye color, these are 2 examples where there are many genes interacting with each other to actually determine a phenotype. So this is going to increase greatly the number of possible phenotypes, and it's going to allow for what appears to be more blending because it is so many different genes that are combining with one another. So the image that you see below sees how the interaction between different numbers of phenotypes allows for this great variation and distribution in terms of those phenotypes. And there's no longer set ratios that you can look at as there is with the monohybrid and the dihybrid cross. All right, now on to gene linkage. So linkage is an important concept that looks at how it is that genes travel together. So Morgan, Thomas Hunt Morgan, was a scientist who worked at Columbia, and he founded a very famous lab called the Fly Lab, and he had his crews called the Fly Boys. And his lab and his students were really set the foundation for modern genetics and being able to link together some of the ideas of inheritance, which certainly Mendel was important in being able to first quantify, but now actually being able to study some of the more complex interactions of genes and seeing how genes are actually physically located on particular chromosomes. Now, flies are a great model organism for genetics, because fruit flies are animals, and so obviously as animals they're going to share a number of genes in common with all other animals, including humans. They are going to have inheritance patterns which are going to be a lot closer to humans. So it's better to study an animal if you're trying to understand how it is that human genetics work. But flies are great because they have 4 chromosomes. So unlike plants, which may have hundreds of chromosomes, unlike humans, which have 23, flies only have 4. So it's easy to be able to figure out exactly what each of those chromosomes do and which genes are located on which chromosomes, because you just have less choices to begin with. Additionally, flies are beneficial because they are small. They don't need a lot of care in order to keep them alive. They are fine living in small spaces, and they grow quickly. They have a very short life cycle, and their lifespan is short. So in terms of genetics, you need to wait for generations, and so you want something that will grow and reproduce very quickly. So in this, since fruit flies only have 4 chromosomes, it was easy to determine sex linkage. And so it was here in fruit flies that we determined the X chromosome and how gender is actually going to be inherited through that X and XY combination. And also was found that there are many other chromosomes that are located, many other genes that are located on that actual chromosome. The X chromosome is very big, Y chromosome is little. This is an example showing a close-up here of the size comparison of the X versus the Y chromosome. And you can see here the map of the Y chromosome, and as you can see, the Y chromosome doesn't really do a whole lot in terms of life-sustaining properties. So one of the really important traits that was tracked by Morgan and his lab was looking at eye color. And eye color was found to be a mutation located on the fruit fly's X chromosome. So there was actually a gene that was associated with the color of the fruit fly's eye, and it was sex-linked. So this is great because eye color is obviously very visible, so it's easy to be able to examine this particular phenotype. And so this was one that was picked up on as a really a breakthrough in being able to study inheritance patterns once it is that they found this particular mutant. How is it that they figured out that it was actually associated with the X chromosome? Well, they looked first and they took the white-eyed males that they had, they crossed those with red-eyed females, and we see all of the resulting offspring are going to have red eyes. Now, we know that they would be heterozygous because based on the Mendelian inheritance patterns, what we should see is that the dominant trait shows up in the F1 generation, and we should see the reappearance in 1 in 4 of those offspring of seeing white eyes. But when they looked at the offspring, they found that all of the female offspring in the F2 generation had red eyes. And again, not looking just at 4 flies, but looking at thousands of flies and thousands of offspring, there were 0 with red eyes. However, there were a 50-50 chance of the males having white eyes. So we had half of the males with white eyes and half of the males with red eyes. So that's what first raised some red flags that the data didn't necessarily match up with their previous understanding of the inheritance patterns for something like this, which is a simple dominant recessive. So the reason for that is considering what it is that the male is passing on. If the male contains an X chromosome and that W represents the white-eyed allele versus the W+, which is the wild type, but it's going to produce the red eyes, in that F1 generation, all the individuals are getting the wild type on that X chromosome from the mother. And the offspring were getting, if they were male offspring, they were getting a Y chromosome which didn't contain that gene. If they were a female offspring, then they were getting that mutated white version on their X chromosome. So in the F1 generation, they aren't actually all the same. We have females. The females are all going to be carriers of that white allele, and all of the males are going to be the wild type. They're going to pass on the allele for red eyes. So when they reproduce with each other, all of those, as we look at all of the offspring, If they are female, that means that the father is passing on a wild type, and so those females that are going to result are going to have at least one copy of that red trait. Those, for the males that are going to be born, the males are getting the Y chromosome from the dad, and because they're getting the Y chromosome from the dad, that makes them male. So that means it's up to the mother. What is the mother passing on? And the mother, again, has a 50-50 chance as to whether the mother would pass on the allele for red eyes or the allele for white eyes. And so that's how we get that ratio on the right for the males of a 50-50 chance of them having white eyes. So as we look at the possible transmission situations for things that are sex-linked. So this is the extension of what it is that Morgan's team had initially discovered. So in the case where we have an affected male, the affected male will pass on that trait to their female, to their daughters. So the daughters can be carriers. The males, any males are going to be unaffected because the affected male cannot pass on that disease to, or that trait to his sons. The sons are all going to get the normal trait from their mother. If we have a carrier mother and an unaffected male, in this case, none of the daughters are going to be affected. We're going to see a possibility that a 50-50 chance of the daughters being a carrier, and we're going to see a 50-50 chance of the males being either unaffected or affected, depending on which of the alleles was passed on from the mother. Finally, if we have an affected father and we have a carrier mother, in this case, the 50-50 chance again for the males to be affected or unaffected, depending on which of those alleles was passed on by the mother. For the females, they're definitely getting one copy of the trait from their dad, and the question then is whether they get that trait from their mom. So we have a 50% chance if they got the normal one from their mom, then they're going to be a carrier. If they get the gene of interest from the allele of interest from their mother, then they're going to have the disease. So as we look at that, for a male to get the disease, then he needs to get that trait from the mother. So the mother needs to be a carrier. For a female to get the disease, they need to get the trait both from their dad and from their mom. So that's why it's going to be more difficult, and we're not going to see as often a scenario where the mother is going to pass on that, where parents will pass on a sex-linked trait to their daughter. So now on to gene linkage. Gene linkage is going to be a case when we have two traits that are both located on genes that are on the same chromosome. So because they're on the same chromosome, they should travel together. There are scenarios, however, due to crossing over, that's going to cause those traits to actually separate from each other and travel independently. If we look at the example, here we have two traits. We have a trait for body color, so we have gray body on the left, black body on the right. We have also the wing type. We have normal wings on the left, and we have vestigial wings on the right. So the individual on the left, the mother here, is going to have both of the wild type or normal traits, and the one on the right, the male on the right, these are both recessive. So what we would expect is that these things are going to travel together. So we would see that the father is passing on these recessive traits together, and the mother is passing on these dominant traits together. So as we look at the numbers, we see that the data on the left shows individuals that are identical to the parents. So we see gray normal wings, 965 of those, and those with black and vestigial wings, like the father, 944 of those. But then there are two other offspring that appear, those that have a combination of the maternal and paternal traits, gray with vestigial wings and black body with normal wings. So neither of those appeared in the parent, and if those traits are actually located on the same chromosome, then they should have traveled together. But what those actually are true is that those are the product of a recombination event. So a recombination of DNA that resulted from the fact that crossing over occurred somewhere in between, as you can see in the picture, somewhere in between those two genes on the chromosome. So if the red versus the peach there was representing maternal and paternal chromosomes, when crossing over occurred during meiosis, that actually swapped, and you can see where the B plus and the B did separate from the VG plus and the VG there on those chromosomes. So any individuals who got those recombined chromosomes would actually end up with a new combination of traits as a result. Now, the percentage of recombination is something that we can analyze to determine how close or how far apart those particular genes are on the chromosome. So here, if you add up 206 and 185, that makes 391 in total. So out of 2,300 total offspring, that was 17%. And if we can compare and see, the higher that percentage is, the further away those genes are actually on a particular chromosome. So we can create something called a linkage map, which is going to map out on a chromosome how far apart particular genes are. So again, we're talking about early 20th to mid 20th century technology here. There was not the ability to map out and actually determine the sequence of genes. We didn't have a technique to be able to find out specifically where on a chromosome a gene was, but they could be mapped out by looking at the recombination of genes on the chromosome. So if we have a chromosome that has 100 genes on it, by looking at inheritance patterns 2 at a time, you can actually map out and figure out where those genes are located in relationship to each other. What you should be looking for in any case when you see data to know if something is linked or not. So remember, if independent assortment occurs, then we should see something like a 3-to-1 ratio, or a 9-to-3 to 3-to-1 ratio, in this case a dihybrid cross. We don't see that's the case here. We see that there is largely a predominance of 2 particular genotypes, which are the parental combinations of phenotypes, and then we see this smaller percentage or smaller number of those recombined phenotypes. So in any case, when you see if the genes are on different chromosomes, an even split, but if the genes are on the same chromosome and parental alleles are linked together, then if they are very closely linked, it's going to be close to 1-to-1 and 0-to-0, but as they are further away on the chromosome, you're going to see a predominance, a 50-50 split of the parental, and then a small but increasing number of those recombined individuals. So this is an example showing gene linkage for a few examples of some mutations that were found and tracked in fruit flies, and the position that they are relative to each other on this particular chromosome. A few final notes here on non-Mendelian inheritance. So other cases in which don't follow that dominant recessive or independent assortment patterns that we expect in Mendel's pea plants. So one is the fact that we have genes that are inherited through odd ways. So those that are inherited matrilineally, so those are inherited only through the mother, and that's going to be the mitochondria and the chloroplasts. Those things are because they are located physically in the egg, there's never a recombination of maternal and paternal DNA in those. So all of your mitochondria come from your mother, and only your mother, not at all from your father. So there is no recombination of DNA there. In males, this is in the nucleus, but the patrilineal inheritance pattern here is the fact that the Y chromosome is inherited only through the father. And so Y chromosomes are things that can inform us a lot about, well, in both cases, mitochondria and Y chromosomes can be useful to look at to determine some larger evolutionary trends. So, for example, human migration can be looked at, and you can see these are different versions, different alleles on the Y chromosome, and areas different populations, how much of those do they express? And so you can see, for example, that the numbers in Western Europe and how much of that particular red mutation appears in the Y chromosome of men there. And so as you look and you can see some of those similarities, as the pie chart here looks more similar, that gives you additional genetic evidence saying that these people would have shared a common ancestry, even though they might now be physically located in different parts of the world. Additionally, epigenetics has an impact on inheritance patterns, and this is referring to a trait and its inheritance that's not determined strictly by DNA. So there's genomic imprinting, so the expression or non-expression of a particular gene as it's inherited from one parent or the other. And also methylation. So methylation is going to be the activation or inactivation of a particular gene by adding on a methyl group or a carbon with 3 hydrogens physically onto the DNA, which either makes it more difficult or easier for it to be transcribed. Finally, environment obviously influences gene expression. So a couple of examples here. We see that flower color in the hydrangea is dependent on pH, so a change in the soil pH impacts the expression of the gene, which then changes obviously the flower color. And then also the seasonal basis, so the change for the Arctic hare from brown to white depending on winter and summer, that again is a change in gene expression that occurs based on the temperature outside. So there's this brings us back to the idea of nature versus nurture, and how much of humans are going to be a product of genes. So our DNA is important, but that DNA does interact with its environment, and it is going to be influenced by a lot of things that are both in and out of our control. And so therefore we are neither beholden to our genes, nor can we fully overcome any limitations within our genes, and it's really a combination of the two working with each other that really determine exactly who it is that we are. Finally, it's important to ensure that the science becomes separated from culture and bias. So there are lots of examples from earlier in the 20th century of how genetics was used to be able to explain the differences between individuals, particularly as we look at different races and nationalities. And it was science was used for much of the 20th century as a way to be able to discriminate, to say that one group of people is better than the other, and using a scientific evidence as a way to be able to support that. So scientists need to be careful that what they discover and what they talk about, the research that they publish, that can't be misconstrued and used by others as a way to be able to perpetuate hatred and bias. With that, if you have any questions, please let me know.